Now we are discussing about genetic code. So we know that during replication and during transcription, what was going on? Nucleic acid was copied to form another nucleic acid. And it was easy to understand because of the process of complementary. Now, what is translation? Translation means you have to convert the information of nucleic acid, means from the polymer of nucleotides to make polymer of amino acid. The translation means sequence of nitrogenous base which is present on messenger RNA has to be changed into the protein language, means sequence of amino acid. So first of all, this is the main thing. Now, main thing is this key we are finding no complementary is present between the nucleotide and amino acid. We know there is no complementary between nucleotide and amino acid, and neither we can draw it also. But there is evidence that if you change a nitrogenous base, amino acid also change. So there are sufficient evidences there to support that a change in nucleic acid is responsible for change in amino acid also. It means there is some code is there. So this led to the proposition of genetic code, which could direct the sequence of amino acid. Now, up till here, I hope you are getting it. Now, if you know this, to know the structure of DNA, it was very much challenging task for the biologist of that era. If determining the biochemical nature of genetic material and to know the structure of DNA was exciting, the proposition and deciphering of genetic code was also very, very challenging. And you require scientists from different streams. We are using the scientist physics person also, we are using organic chemistry person also, we are using biochemist person also, and genetics person also. And the first person was George Gamow, who was actually a physics, who told कि if there are only type of nitrogenous bases, if we see, there are only 4 types of nitrogenous base, and amino acid, if we see, the types of amino acid is only 20 type. So there should be some relationship between them. 1:1. If I say each codes for 20 amino acid, not possible. Set of 2 means 16. This is for 20. This is also not possible. Then set of 3. Yes, this is possible. For 20 amino acid, this is possible. So he told ki if there are only 4 bases and if they have to code for 20 amino acid, there should be some combination. And what he told, it should be made up of 3 nucleotides. Means set of 3 is one genetic code which code for one amino acid. And this was very bold proposition because it generates 64 codon more than required. So first scientist, please learn, first scientist George Gamow, who gave the bold proposition, the permutation combination of 4 raised to power 3 will code for one amino. Matlab, this will generate the codons. So if suppose I give you this question, suppose we are giving it, this is a messenger RNA, which is starting from 5' AUG. Suppose I'm writing it, this way sequence, I'm continuously writing it. Then how to know it? You should know ki set of 3. This is one set. Set of 3 is actually one codon. Set of 3 means one codon. And one codon codes for one amino acid. That is why we are saying ki codons are triplet. That is why we say codons are triplet, means set of 3 nitrogenous bases means one codon, and one codon means one amino acid. Now, next scientist was Hargobind Khurana, and he developed a chemical method to prepare RNA molecule with defined combination of bases, either homo or copolymer. Now, what does it mean? Homopolymer example, suppose you write only UU. This is homopolymer. Or suppose AA. Any set only one time repeated, that is homopolymer. One type. Copolymer means suppose I'm using CU set, this way. Or suppose AC, whatever it is. The codon, we know it is triplet. So this is a set of 3. Set of 3. Set of three. This way. Are you getting it? So he prepared RNA in defined combination of bases by using DNA as a template. The first person was Hargobind Khurana. First person was George Gamow. And now second person was Hargobind Khurana. Now come to the third person. Third person was Nirberg. Nirberg, what he did, he prepared protein without using cell to decipher the genetic code. Cell-free system means he does not use cell. Without using cell, he has prepared the protein to help the deciphering the code, means triple U codes for phenylalanine, for example. This is the way how deciphering is done. Now, next scientist was Severo Onko. And what he did, he has prepared RNA, in defined combination of bases without using DNA. He prepared RNA with defined sequence in template independent manner, means without using DNA. To give a respect to him, we are saying the name of the enzyme is Severo-oncoenzyme, and actually which enzyme? Enzyme was polynucleotide phosphorylase. So four scientists, please learn. First, Gamow, George Gamow, physics, permutation combination. Hargobind Khurana, making RNA, homo and copolymer. Nirberg, cell-free system, protein synthesis. Severo-onco, RNA without using template. And finally, this is the way how checkerboard is done. So this is the first position. Okay, everywhere we have to put in this box. Suppose this is the first U only. Check first position. Okay, C only first position. Then second position is U. So in this line, if you see everyone, second position is U only. Second position is U. And this is the third position. Third position is U here. C here. This way. A. And this is G. So this is, you have to just learn it. If possible, it is good. If not, then don't take stress, because in the exam, they will not ask you that much question. They'll ask you only which is the starting codon. So you should know AUG is a start codon, which codes for methionine. Which is stop codon? Stop codons are three: UAA, UAG, and UGA. These are stop codons. Clear? You should learn it. So total, if you see, out of 64 codon, 3 are stop codon. It means 61 codon code for amino acid, and 3 codons do not code for any amino acid. So they are stop codon. Which are stop codon? Once again, write UAA, UAG, and just reverse now UGA. Now, codon should be read in the continuous manner. There should be no full stop, no comma, no punctuation, nothing is there. It is universal. Because it is universal, that is why in the biotech we are using this phenomena only. If you know, from bacteria to human, everywhere triple U means phenylalanine. Though exceptions are there. Exceptions are present in mitochondria and in some protozoans. Now, if you see one codon, code for only one amino acid. Code for one amino acid. Is it clear? It code for one amino acid. Means I will say codon are specific. Codon are specific. They code for one amino acid. But see the line of what I'm writing. One amino acid can be coded by many codon. Can be coded by many codon. It means codon degenerate. Codon degenerate. Write this way, it will be helpful for you, because this is a question they can twist you. Some amino acid are coded by more than one codon. We say codon degenerate. I told you AUG is a starting codon. So it codes for methionine and it's a starting codon. And these three are your stop codon. Clear? So genetic code, this much is clear. And if you have any doubt, then ask. Then we'll move further. Now, next topic is mutation. See, first of all, relationship between genes and DNA is best understood by mutation studies. Effect of large deletion and rearrangement are easy to comprehend. It may result in the loss or it may result in the gain of a gene and as a result function. Very best example of point mutation is sickle cell anemia. In sickle cell anemia, change is coming in the single base only. One base is changed because of which amino acid is changed. So what happened? Example of point mutation is sickle cell anemia where we change a single base pair and especially in the beta chain. Beta globin change result in the change of amino acid from glutamic acid to valine. Glutamic acid in the healthy and after change it becomes valine and as a result disease is known as sickle cell anemia is made. The effect of point mutation that insert or delete a base in structural gene can be understood if suppose we are taking a, this is also proof, yes, genetic codes are triplet because suppose these are the three words you're taking it. And if you add in B in between, so you see after the moment you add this one letter, your reading frame is disturbed from the point you have done the insertion. If you do the insertion of 1 or if you do the insertion of 2, your reading frame is disturbed. But if you insert 3 letters, your frame is not disturbed, only one amino acid is extra. So same with the addition and same with the deletion. So what is conclusion? Conclusion is insertion or deletion of 1 or 2 will change the reading frame from the point of insertion or deletion. It means reading frame is disturbed and such type of mutation is known as frame shift mutation. Such type of mutation is known as frame shift mutation. If you are inserting, then frame shift insertion mutation, or if you're deleting, then frame shift deletion mutation. But if suppose we do insertion of 3 or its multiple, if you do the insertion or deletion of 3 or its multiple, it means only 1 or 2 amino acid will be inserted or it will be deleted. Reading frame will remain undisturbed. So this proof, this insertion of 3 or its multiple proof, yes, genetic code is triplet in nature. It is triplet in nature. So up till here, just read. If you have any doubt, then ask. See, transfer RNA is also known as adapter molecule. It is also known as soluble RNA. Please understand, because in our NCRT, one where we are studying snRNA, if you remember, we studied in the transcription area, that is small nuclear RNA, which is used for splicing. And this is sRNA, soluble RNA. Now, when the proposition of code was done, so before the very beginning of the proposition of code, it was clear to Francis Crick that there should be some mechanism who can read the genetic code and can join the amino acid. Because amino acid don't have any structural specialties to read the code directly. So someone should be there who can read the code and who can bring the amino acid. So he is the one who postulated the presence of adapter molecule, which can read the code and on the other hand, it can bind to amino acid. So that time, understand it, that time it was known as soluble RNA. The transfer RNA then called, that time it was called as soluble RNA, was known before the genetic code was postulated. But its role as an adapter RNA was known later on. Now, if you see in the transfer RNA, there is anti-codon loop is present in transfer RNA. It is having amino acid attachment site at the third end. Please learn properly. Amino acids are attached at third end. Fifth and no amino acid is attached. And if you see codon are present, where codons are present on messenger RNA. So if suppose codon is coming from 5' to 3', anticodon has to come from 3' to 5'. So please check carefully, is it coming or not? See here, start from the 3'. See this way and start. And yes, it is coming from 3' to 5'. So anticodon has to be read from 3 dash to 5 dash. So if suppose they give you question and they give you 5 dash to 3 dash, and suppose they give A, C, G or A, U, U. So it means this is talking about codon and this is messenger RNA, and your amine transfer RNA should be read from 3 dash to 5 dash. So transfer RNA will have anticodon, which is UGC and UAA. Clear? So this is anticodon, and anticodon are present on transfer RNA. Please learn properly. Anticodons are present on transfer RNA. Don't confuse yourself. And to learn properly, the transfer RNA is having anticodon loop. Transfer RNA is having anticodon loop, means base complementary to the codon. It has amino acid acceptor and which one? Third end, which will bind to the amino acid, and every transfer RNA is specific for each amino acid. For starting, there is initiator transfer RNA. Okay, for starting, there is initiating transfer RNA, and learn properly, there is no transfer RNA for stop codon. Now, 2D structure and 3D structure. Secondary structure means 2D structure of transfer RNA is clover leaf-like, but 3D structure means actual structure is inverted L. So learn it. 2D structure is clover leaf-like, and 3D structure means actual structure is inverted L. So I think so this much topic might be easy for you. Just read and see carefully if you have any doubt.